Welcome friends, welcome to the Knowledge Club Online. Today I am going to discuss about the initiation of eukaryotic replication. Initiation of eukaryotic DNA replication is the first stage of DNA synthesis where the DNA double helix is unwound and an initial priming event by DNA polymerase alpha occurs on the leading strand. The priming event on the lagging strand establishes a replication fork. Priming of the DNA helix consists of synthesis of an RNA primer to allow DNA synthesis by DNA polymerase alpha. Priming occurs once at the origin on the leading strand at the start of each Okazaki fragment on the lagging strand. One replication is initiated from specific sequences called origins of replication, and eukaryotic cells have multiple replication origins. To initiate DNA replication, multiple replicative proteins assemble on and dissociate from these replicative origins. The individual factors described below work together to direct the formation of the pre-replication complex, a key intermediate in the replication initiation process. Association of the origin recognition complex ORC with a replication origin is required to recruit both cell division cycle 6 protein CDC6 and chromatin lysine and DNA replication factor 1 protein CDT1, which initiate the assembly of the pre-RC. Both CDC6 and CDT1 proteins associate with the already bound ORC independently from each other. The ORC, CDC6 and CDT1 together are required for the stable association of the mini-chromosome maintenance MCM 2 to 7 complex proteins with replicative origins during G1 phase of the cell cycle. First, I discuss the pre-replicative complex. Diagram of the formation of the pre-replicative complex transforming into an active replisome. MCM 2 to 7 complex loads onto DNA at replication origins during G1 and unwinds DNA ahead of replicative polymerase. CDC6 and CDT1 during bring MCM complexes to replication origin. CDK or DDK dependent phosphorylation of pre-replicative proteins leads to replisome assembly and origin firing. CDC6 and CDT1 are no longer required and are removed from the nucleus or degraded. MCMs and associated proteins, genes and CDC45 unwind DNA to expose template DNA. At this point, replissome assembly is completed and replication is initiated. P represents the phosphorylation. Eukaryotic origins of replication control the formation of a number of protein complexes that lead to the assembly of two bidirectional DNA replication forks. These events are initiated by the formation of the pre-replication complex pre-RC. At the origins of replication. This process takes place in the G1 stage of the cell cycle. The pre-RC formation involves the ordered assembly of many replication factors including the origin recognition complex ORC, CDC6 protein, CDT1 protein and mini-chromosome maintenance proteins MCM 2 to 7. Once a pre-RC is formed, activation of the complex is triggered by two kinases, cyclin dependent kinase 2 CDK and DBF4 dependent kinase DDK that help transition the pre-RC to the initiation complex prior to the initiation of DNA replication. This transition involves the ordered assembly of additional replication factors to unwind the DNA and accumulate the multiple eukaryotic DNA polymerase around the unwound DNA. Next, we come to the origin recognition complex. The first step in the assembly of the pre-replication complex (pre-RC) is the binding of the origin recognition complex (ORC) to the replication origin. In late mitosis, CDC6 proteins joins the bound ORC followed by the binding of the CDT1 protein. ORC, CDC6 and CDT1 are all required to load the 6 protein mini chromosome maintenance MCM227 complex onto the DNA. The ORC is a 6 subunit ORC1p6 protein complex that selects the replicative origin sites on DNA for initiation of replication, and ORC binding to chromatin is regulated through the cell cycle. Generally, the function and size of the ORC subunits are conserved throughout many eukaryotic genomes, with the difference being which ORC unit actually contacts the DNA. The most widely studied origin recognition complex is that of Saccharomyces cerevisiae or yeast, which is known to bind to the autonomously replicating sequence ARS. The replication origins of higher eukaryotes are made up of similar AT-rich regions. The yeast cerevisiae ORC interacts specifically with both the A and B1 elements of yeast origin of replication, spanning a region of 30 base pairs. The binding to these sequences requires ATP. The S cerevisiae ORC requires the 5 largest subunits ORC1, ORC2, ORC3, ORC4 and ORC5 to recognize DNA, 4 of which ORC1p, 2p, 4p and 5p remain in close contact with the origin. The ORC1 and ORC5 subunits are known to interact with ATP, but only the interaction between the ORC1 subunit and ATP are required for DNA binding. The S cerevisiae ORC1 subunit also hydrolyzes ATP, however ATP hydrolysis is not required for DNA binding. Once the ORC is bound to the origin, the complex is retained in an ATP bound state and the ATP hydrolysis is reserved for a downstream step in initiation. When the ORC binds to DNA at replication origins, it then serves as a scaffold for the assembly of other key initiation factors of the pre-replicative complex, which include CDC6, CDT1 and mini chromosome maintenance MCM227 complex proteins. The pre-replicative complex assembly during the G1 stage of the cell cycle is required prior to the continuation of DNA replication during the S phase. The recognition of mammalian ORC is consistent with the removal of at least part of the complex from the chromosome at metaphase. The ORC1 associated with chromatin is released during mitosis. The removal of ORC could serve to eliminate the pre-replicative complex formation prior to the completion of metaphase. Now we come to the CDC6 protein. What is CDC6 protein? Binding of the cell division cycle CDC6 protein to the origin recognition complex ORC is an essential step in the assembly of the pre-replication complex pre-RC at the origins of replication. CDC6 binds to the ORC on DNA in an ATP-dependent manner, which induces a change in the pattern of origin binding that requires ORC10 ATPase. CDC6 requires ORC in order to associate with chromatin and is in turn required for the mini chromosome maintenance proteins MCM227 to bind to the chromatin. The ORC CDC6 complex forms a ring-shaped structure and is analogous to other ATP-dependent protein machines. The levels and activity of CDC6 regulate the frequency with which the origins of replication are utilized during the cell cycle. Now the CDT1 protein is a fission yeast and xenopus chromatin lysine and DNA replication factor 1 CDT1 protein is required for the lysining of chromatin for DNA replication. CDT1 is essential for DNA replication and performs its role during the formation of the pre-replicative complex by loading the mini chromosome maintenance proteins onto the chromosome. CDT1 has been shown to associate with the C-terminus of CDC6 to cooperatively promote the association of CMC proteins to the chromatin. CDT1 activity during the cell cycle is tightly regulated by its association with the protein geminin, which both inhibits CDT1 activity during S phase in order to prevent re-replication of DNA and prevents it from ubiquitation and subsequent proteolysis. Now I come to the mini chromosome maintenance protein complex. The assembly of the mini chromosome maintenance MCM proteins function together as a complex in the cell. The assembly of the MCM proteins onto chromatin requires the coordinated function of the origin recognition complex ORC, CDC6 and CDT1. Once the MCM proteins have been loaded onto the chromatin, ORC and CDC6 can be removed from the chromatin without preventing subsequent DNA replication. This suggests that the primary role of the pre-replication complex is to correctly load the MCM proteins. MCM proteins support roles both in the initiation and elongation steps of DNA synthesis. Each MCM protein is highly related to all others, but unique sequences distinguishing each of the subunit types are conserved across eukaryotes. All eukaryotes have exactly 6 MCM proteins, along so that each fall into one of the existing classes MCM 2 to 7, which suggests that each MCM protein has a unique and important function. Mini chromosome maintenance proteins have been found to be required for DNA helicase activity, and inactivation of any of the 6 MCM proteins prevents further progression of the replication fork. This is consistent with the requirement of ORC, CDC6 and CDT1 function to assemble the MCM proteins at the origin of replication. The complex containing all 6 MCM proteins creates a hexameric dough-like structure which a central cavity. The helicase activity of the MCM protein complex raises the question of how the ring-like complex is loaded onto the single-stranded DNA. One possibility is that the helicase activity of the MCM protein complex can oscillate between an open and a closed ring formation to allow single-stranded DNA loading. Along with the mini chromosome maintenance protein complex helicase activity, the complex also has associated ATPase activity. A mutation in any one of the 6 MCM proteins reduces the conserved ATP binding sites, which indicates that ATP hydrolysis is a coordinated event involving all 6 subunits of the MCM complex. Studies have shown that within the MCM protein complex are specific catalytic pairs of MCM proteins that function together to coordinate ATP hydrolysis. For example, MCM6 but not MCM3 but not MCM6 can activate MCM6 activity. These studies suggest that the structure for the MCM complex is a hexamer with a MCM3 next to MCM6. Contribute to the formation that allows ATP binding and hydrolysis, and the mixture of active and inactive subunits create a coordinated ATPase activity that allows the MCM protein complex to complete ATP binding and hydrolysis as a whole. The nuclear localization of the mini chromosome maintenance proteins is regulated in budding yeast cells. The MCM proteins are present in the nucleus in G1 stage and S phase of the cell cycle, but are exported to the cytoplasm during the G2 phase and S phase of the cell cycle, but are exported but are exported to the cytoplasm during the G2 stage and M phase. A complete and intact 6 subunit MCM complex is required to enter into the cell nucleus. In S. cerevisiae, nuclear export is promoted by cyclin-dependent kinase CDK activity. MCM proteins that are associated with chromatin are protected from CDK export machinery due to lack of accessibility to CDK. Now I come to the initiation complex. During the G1 stage of the cell cycle, the replication initiation factors, origin recognition complex ORC, CDC6, CDT1 and mini chromosome maintenance MCM protein complex bind sequentially to DNA to form the pre-replication complex. At the transition of the G1 stage to the S phase of the cell cycle, S phase specific cyclin dependent protein kinase CDK and CDC7 DBF4 kinase DDK transform the pre-RC into an active replication fork. During this transformation, the pre-RC is deassembled with the loss of CDC6, creating the initiation complex. In addition to the binding of the MCM protein cell division cycle 45, CDC45 protein is also essential for initiating DNA replication. Studies have shown that MCM is critical for the loading of CDC45 onto chromatin, and this complex containing both MCM and CDC45 is formed at the onset of the S phase of the cell cycle. CDC45 targets the MCM protein complex, which has been loaded onto the chromatin as a component of the pre-RC at the origin of replication during the G1 stage of the cell cycle. CDC45 protein. Cell division cycles 45, CDC45 protein is a critical component for the conversion of the pre-replicative complex to the initiation complex. The CDC45 protein assembles at replication origins before initiation and is required for replication to begin in Saccharomyces cerevisiae and has an essential role during elongation. The CDC45 has central roles in both the initiation and elongation phases of chromosomal DNA replication. CDC45 associates with chromatin after the beginning of initiation in the G1 stage and during the S phase of the cell cycle. CDC45 physically associated with MCM5 and displays genetic interaction with 5 of the 6 members of MCM gene family and the ORC2 gene. The loading of CDC45 onto chromatin is critical for loading other various replication proteins, including DNA polymerase alpha, DNA polymerase epsilon, replication protein A, RPA, and proliferating cell nuclear antigen PCNA onto chromatin. Within a xenopus nucleus-free system, it has been demonstrated that CDC45 is required for the unwinding of plasmid DNA. The xenopus nucleus-free system also demonstrates that DNA unwinding and tight RPA binding to chromatin occurs only in the presence of CDC45. Binding of CDC45 to chromatin depends on CIB CDC28 kinase activity as well as functional CDC6 and MCM2, which suggests that CDC45 associates with the pre-RC after activation of S-phase cyclin-dependent kinase, as indicated by the timing and the CDK dependence. Binding of CDC45 to chromatin is crucial for commitment to initiation of DNA replication. During S-phase, CDC45 physically interact with MCM proteins on chromatin. However, dissociation of CDC45 from chromatin is slower than that of MCMs, which indicates that the proteins are released by defined mechanisms. Next is genes. The 6 mini-chromosome maintenance proteins and CDC45 are essential during initiation and elongation for the movement of the replication forks and for unwinding of the DNA. Genes are essential for the interaction of MCM and CDC45 at the origins of replication during initiation and then at DNA replication forks as the replisome progresses. The genes complex is composed of 4 small proteins: SID5, CDC105, PSF1, CDC101, PSF2, CDC102, and PSF3, CDC103. Genes represents Go, Ichi, Nai, Shan, which means 5123 in Japanese. Next is MCM10. MCM10 is essential for chromosome replication and interacts with the mini chromosome maintenance 227 helicase that is loaded in an inactive form at origins of DNA replication. MCM10 chaperones that catalytic DNA polymerase alpha and helps stabilizes the polymerase. Next is DDK and CDK kinase. At the onset of S phase, the pre-replicative complex must be activated by 2 S phase specific kinases in order to form an initiation complex at an origin of replication. One kinase is the CDC7 DBF4 kinase called DBF4 dependent kinase DDK, and the other is cyclin dependent kinase CDK. Chromatin binding assays of CDC45 in yeast and xenopus have shown that a downstream event of CDK action is loading of CDC45 onto chromatin. CDC6 has been speculated to be the target of CDK action because of the association between CDC6 and CDK and the CDK dependent phosphorylation of CDC6. The CDK dependent phosphorylation of CDC6 has been considered to be required for entry into the S phase. Both the catalytic subunits of DDK and CDC7 and the activator protein DBF4 are conserved in eukaryotes and are required for the onset of S phase of the cell cycle. Both DDK and CDC7 are required for the loading of CDC45 onto chromatin origins of replication. The target for binding on the DDK kinase is the MCM complex, possibly MCM2. DDK targets the MCM complex and its phosphorylation leads to the possible activation of MCM helicase activity. Next I come to the DBP11, SID3 and SID2 proteins. SID3, SID2 and DBP11 interact with many replication proteins. SID3 and CDC45 form a complex that associated with pre-RC at the early origins of replication even in the G1 phase, and with the latter origin of replication is the S phase in a mutually MCM dependent manner. DBP11 and SID2 interact with polymerase epsilon, and cross-linking experiments have indicated that DBP11 and polymerase epsilon co-precipitate in the S phase and associated with replication origin. SID3 and SID2 are phosphorylated by CDK, which enables the two replicative proteins to bind to DBP11. DBP11 had two pairs of BRCA1 C-terminus BRCT domains, which are known as the phosphopeptide binding domains. The N-terminal pair of the BRCT domains binds to phosphorylated SID3, and the C-terminal pair binds to phosphorylated SID2. Both of these interactions are essential for CDK-dependent activation of DNA binding in yeast. DBP11 also interacts with genes and participates in the initiation and elongation steps of chromosomal DNA replication. Genes are one of the replication proteins found at the replication forks and forms a complex with CDC45 and MCM. These phosphorylation depends interactions between DBP11, SID2 and SID3 are essential for CDK-dependent activation of DNA replication, and by using cross-linking reagents within some experiments. A fragile complex was identified called the preloading complex, pre-LC. This complex contains polymerase epsilon genes, SID2, and DPB11. The pre-LC is found to form before any association with the origins in a CDK dependent and DDD dependent manner, and CDK activity regulates the initiation of DNA replication through the formation of pre-LC. This lecture may be helpful to you. Thank you.